All right. Chapter 4, we're looking at DNA and RNA. We're looking at nucleotides and polynucleotides. This is a picture of a designer DNA molecule. Now, this is not something you would naturally find, okay? I mean, look at it. It looks like a bunch of triangles, right? Designer DNA is something fairly pretty new. Think of even about different base pairs. Imagine if you could alter a base pair and get it to replicate. What if you could have a synthetic base pair and actually get it to replicate in vivo? What do you think that might do?
Violate some code of ethics.
Violate some code of ethics? Okay, all right. I tell you what, let's talk about that a little bit in just a little bit. Okay, let's talk about that in just a little bit. But this is a T2 bacteriophage. This is a virus that is in your book, and it infects E. coli. And this actually came from Hershey and Chase's original experiment of actually discovering things about DNA and how DNA is put together, essentially. And the book in chapter 4 talks a lot about the history of DNA and RNA. How many people have started reading chapter 4? Anybody? A little bit, okay, almost no one. All right, well, I'm gonna hold you to it. But there's a lot of information in chapter 4. Some of this will be review for you guys, and some of this will not be review. How many people have had quite a bit of this in other courses? Would you say? What's that?
I have not read this.
That's okay.
What's it about?
Quite a bit of what?
Nucleic acids, DNA, RNA.
Oh, yeah.
You've had plenty, yes,
Yeah.
already?
Decent amount,
You've
not
had
like,
doctors in
it's a lot more.
Through Dr.
complicated.
Zimmerman, right? I mean, mostly. So chapter 4 is entitled Nucleic Acids, okay? Now, so what they did on this one is they ruptured the head of the viron with a osmotic shock. So I'm assuming they put it into salt water and threw it back into fresh water or something like this that caused it to rupture. And this is all the DNA that spewed out of the head of the viron, which is crazy. I mean, you think about like that much material in that one little, what do they call it? A capsid? What do they call the head of the?
I think it's called the capsid.
Yeah.
Isn't it called a capsid?
Sounds like something.
I can't remember, but anyway, it's pretty impressive. How many people have ever precipitated out DNA and actually seen what it looks like? Anybody ever precipitated out and seen what it looks like? Have you ever done onion DNA? Precipitated that out? That's actually pretty easy. Riley, what's it look like?
I mean,
When it comes out into solution, what is precipitated?
It's cloudy. Like I looked at it in a pellet. Like, but I did like my own.
It,
I didn't see anything.
yeah, it's kind of stringy, snotty looking. It's opaque. Yeah, absolutely. And it's really fascinating. I remember the first time I ever precipitated DNA out of onion in college, and I thought it was the coolest thing ever to know that I was actually looking at DNA. Like, you never could think, like, wow, this is really small. How could you ever actually see it? But when you have a lot of it, then you actually can tell what's going on. Difference between RNA and DNA. This comes down to the sugars. DNA is the deoxy version of RNA. We know that RNA can do different things than DNA. DNA will only encode, right? It's only there for storage of genetic information, whereas RNA can do what? What can RNA do? Yeah, so it can translate and store information, both. So we think translation, we think tRNA, right? And the RNA technologies that are being developed now are really just phenomenal. And there's so much going on right now with RNA research. What else? Why phosphates? Why phosphates? Why did our creator choose phosphates? Okay, how many times they can bond? I think that's good. What else?
Aren't they good as leaving groups?
They are good as leaving groups, and therefore they must be
Have great resonance?
resonance, which contributes to stability.
Mm-hmm.
Yeah, absolutely. So think about resonance. We also think about stability of not only the monoanion, but stability of the dianion as well, okay? And those different pKa's. We've never figured out anything that can replace phosphorus in terms of its unique stability. It's phenomenal for what it does. If you look at the phosphodiester linkages between DNA and RNA, they are the same. You still get the same type of polynucleotide sequences, but when we start looking at the differences between DNA and RNA a little bit more closely, we see that there are differences in the base sequences, right? So we have different bases that are used. Which one was discovered first? DNA or RNA? What do you think? Riley says RNA.
I would think RNA, because they named one after a modified version of the other.
Okay? It's actually DNA. DNA was actually the one that was discovered first. RNA was actually not discovered till much later, which is kind of fascinating. I didn't know that, and that's something that I learned from the reading as well. We do think that RNA may have been the first thing that was developed because it can translate and store information both. From an evolutionary standpoint, they think that RNA was first and then DNA. Okay? I have strong feelings about that, and I think that they were both designed for two different purposes. And I think that that's really up to you and your beliefs. The base pairs are all derived from either purine or pyrimidine nitrogen heterocycles, and these are the base heterocycles for each of these. You can buy purines and pyrimidines on the synthetic market, and you can design your own molecules from these. They don't have to be produced by nature. We can produce these synthetically now. We can order these from Sigma Aldrich or other chemical suppliers and do all different types of chemistry with many different nitrogen heterocycles. There are so many nitrogen heterocycles, I don't even have them all memorized, okay? I mean, there are a ton of them, yep. Let's take a look at the ones that we know about so far, and the purines are on the left. You've seen adenine and guanine. Both of those are found in DNA and RNA. The right-hand side, the pyrimidines, some of those found in DNA and RNA like cytosine, but thiamine's only found in DNA, and uracil's only found in RNA. What about this 5-methyl cytosine? Have you seen 5-methyl cytosine before? And where is it found? Okay?
It's created.
Oh, hold on. I know this is part of your research. So hold on. Where do you think it came from? And why do you think it may have happened. Where did it come from? Yeah, from which one?
Is it from
Oh,
cytosine 9?
Okay, so if it came from cytosine, okay, then it simply got methylated. It had a methyl group get put onto it, right? I didn't realize, or I didn't know when 5-methylcytosine was discovered, because I did not learn about the methylated version of this when I was in college. But it was discovered actually initially in the late 1800s and early 1900s, and it wasn't actually identified and studied like extensively until many decades later. But it was actually identified really early on, a lot earlier than I would have expected it to have been identified. But it actually happens naturally. And we've started to understand why this happens, but we don't know exactly the whole story. Okay, Riley, do you want to expand a little on it?
Well, they think it happens in order to deactivate DNA. Attaching the methyl group causes it to become more constricted and less active, but also it cycles, and like 5-methylcytosine can accidentally be made into thymine and uracil, and then has to be replaced by the body and by polymerases. And that also can cause increases mutations that happen before transcription.
Yeah.
And so if it's not caught, you get genetic variation.
That's right. If it's not caught, you get genetic variation. You can also, those mutations can lead to various cancers and so forth. But it does happen naturally. And the study, or the field of study, is called epigenetics. And you should write that down. E-P-I genetics, epigenetics. A friend of mine actually studied epigenetics of various native cultures in Alaska, and found out that DNA methylation happened quite often, or more prevalently in some of these cultures who had different diets, and so that they think that diets can actually result in changes in your genome. Yes, yes. And this whole field of epigenetics encompasses all of this. We didn't think that there was anything that really changed our genetics, while we were living on Earth, that our genes are the same all the time. But we're finding out now that that's not true. Yeah. So really very fascinating field. There's a lot going on in that field. So Riley's wanting to do research in that area for her project for senior year. Okay? Any questions on that? Well, which bases pair up? Which bases pair up?
C and G.
Okay, C and G. What else?
AT.
Adenine, thymine,
And adenine uracil.
and then what?
Adenine and uracil.
Adenine and uracil, and that replaces thymine and RNA. Yeah? Okay. How did they pair up? Should I have you build them with models today and pair them?
Yes,
Yes?
that sounds fun.
That sounds like fun? Probably sounds like a lot more fun than actually talking about DNA and RNA. Do we have enough models to do it? That's the question.
If each person builds a different base, then we could.
Where's our box? Where's our box of it was in here. It may be in the next room over.
Last one I saw it was in the Gen Chem lab.
It was in the Gen
I
Chem
think we're
lab.
using it for.
You wanna go get it? You wanna go try? Yeah? we'll talk a little bit more about what holds these things together, and maybe if you make one base and somebody else makes a different base and you try and put them together, maybe you'll be able to see those areas that interact. Maybe you'll be able to just have a little bit of fun with it. I'll wait until she gets back. I didn't know, I was reading about DNA and RNA being metastable polynucleotides. Anybody know what metastable means?
Very stable.
Okay, think about stable versus metastable, even the word meta.
Is that a placement? Like ortho?
No?
No, it's not.
Well, that's on aromatic rings in
Oh,
organic
okay.
chemistry.
Is that when it's aware that it's stable?
It's aware that it's stable. No?
Is that to do
It's
with,
Facebook
like,
stable.
having stack destabilize itself?
No? No?
It has an Instagram. Meta, sorry.
It has to do with stability. It has to do with stability under different conditions. So it's a conditional stability. So metastable is essentially a conditional stability. DNA and RNA can both be hydrolyzed under acidic conditions, acid catalyzed conditions, which means they can essentially break bless you, they can start breaking apart the bonds between the phosphates. We can go back and look. And so those phosphodiester linkages are susceptible to acid catalyzed hydrolysis conditions. And I didn't know this, but RNA is actually susceptible to base catalyzed conditions, whereas DNA necessarily is not. And I thought that was kind of fascinating. So coming back, metastable means a conditional type stability, and that is important to life because if DNA wasn't stable enough, it wouldn't support life. And if it was too stable, you wouldn't be able to unzip it and replicate.
Okay, so something I was kind of confused about. So it's an acid, deoxyribonucleic acid, and it has a net negative charge. Why does it have a net negative charge? Because in like PCR works because it has a negative charge.
That's right, yeah. So why does it have a net negative charge? I don't know. It clearly has to do with one of, I'm thinking it's one of the ends. One of the ends is going to have a negative charge on a functional group, I think. So let me do a little digging and see what I can find out. But I do know you're right. It does have a net negative charge. I just can't remember why it has it. So clearly, is it the phosphates?
Mm-hmm.
Is it?
Probably.
Yeah, it's due to the bonds created between the phosphorus atoms and the oxygen atoms.
Okay.
That must be it. Yep, that must be it. So, PCR. How does it work by the charge?
It's a negative charge on one side and a positive charge on the other, and so the negative, will move from the negative positive side based on its speed will change by its size.
Okay, so it migrates from one electrode to the other electrode based upon just the attraction of the voltage. Cool. How does that different then between electrophoresis? Because electrophoresis works with charge too, right? So, I mean, they're two different techniques. Do you know the difference? Devin, do you know the difference between those two? Well, we'll figure it out. That's what this class is for. All right, Devin, you found kits?
Yes, intro is using them, so I only grabbed what I
Oh,
could.
intro is using them like right now?
For lab.
Oh my goodness. Okay. Let's do groups then. Okay, so let's group up, and it's probably gonna take a few hits probably to make one base. I might see what we can come up with. You wanna push pause on that? Just stop the video.